of implantation in pregnancy, we've gotten essentially to this point where the fertilized egg, the trophoblast, is getting itself embedded into the maternal endometrial wall. And in primates, it's all the way into that structure, not to the myometrium, but just into the endometrial wall, where it can gain access to blood vessels. In a variety of, let's see, I'm going to go to the next few stages of growth. Where you're going to see further development occurring. There's intimate contact between the syncytial trophoblastic cells and these maternal blood vessels, which sometimes break down and form what are called lacunae. And that's going to be a source of nutrient supply, waste removal for the these early stages of embryonic development. What you're starting to see, though, is differentiation in the inner cell mass and the formation of more specialized cavities. So you go from what used to be the yolk sac here and the amnion, and you start to form what is going to become the coelom here, and this is going to go on and form the chorionic area. Chorion filled with fluid, as does the amnion. Okay, so these are in the next few days. So A here is by 10 days after fertilization. This is 12 days. So even here, you're essentially at the time when, if a menstrual period was anticipated, this is where it would be, this is the region where it would be occurring. Interesting to note a couple of things. Even at this stage, there are a group of cells that sit near the yolk sac at the edge of the embryo. They're called the procordial plate. And that's the region right near there is the region where the precursors for hematopoietic, that is blood cell formation, and for gametopoietic cells, that is the germ stem cells, that's where those are going to develop from. So you've got those precursors starting from outside the embryo proper. So let's go. We've gone through hormonal profiles, so I'm not going to bring that back up again. And I did mention the coordinated production of steroids in the maternal fetal placental unit, where you've got the placenta being a source for a lot of progesterone. Progesterone and pregnenolone being processed by the fetal adrenal cortex, which is different, developmentally different from the adult adrenal cortex, even though it does some of the same things. It does make cortisol and aldosterone from progesterone, but from pregnenolone you get weak androgens, which are going to be further processed by the fetal liver into estrogens, and particularly this one, estriol, which is the major steroid estrogen of pregnancy. So mom is out here, outside this circle. So her organs and tissues, her liver, her adrenals, her gonads are actually outside that circle. But it's a coordinated effect to get the steroids that you see most prominently in during pregnancy, the progesterone and the estrogens, particularly estriol. Okay, let's go back. Get me down this list a little bit. Okay. All right. I wanted to get to this slide. This slide presents the developmental progression during pregnancy in the human or in the primate. And notice that this is the time before an anticipated menses would occur. So all these events down to this stage where you're starting to see the notochord or the nervous system begin to differentiate is all occurring prior to the anticipated normal menses time, the end of the duration of a non-fertile corpus luteum. All that would occur in here. And chorionic gonadotropin is going to begin to rise, particularly after you've got contact here and intimate interaction between the trophoblast, which is making a lot of this chorionic gonadotropin, and the blood vessels in the maternal circulation, which is going to bring it into maternal circulation and into maternal waste products. During the time up to here, between about 3 and 4 weeks, is usually when you're going to get clinical recognition of pregnancy. That's the time when you can begin to ascertain the presence of these cavities within the developing placenta by ultrasound. So this is when clinical recognition would occur. Between this time and this time, you have what's called a biochemical pregnancy that can be detected with chorionic gonadotropin or an evaluation of the steroids, maternal steroids in circulation, because the estrogens and the progesterones are going to start going up in this period of time. Note you've got various systems that develop at different times and in different rates. The central nervous system is beginning to develop down here, and it continues to develop not just to here, but way out beyond that, and probably to the mid-20s in males. And at least the late teens to early 20s in females. So nervous system development is a long-term project. Circulatory development, particularly of the heart, begins early on, even before this clinical recognition, but it extends particularly into these 6th, 7th, and 8th weeks. So this is 2 months of pregnancy. At about the same time frame, you're seeing the limbs develop, arms and the legs, and you're beginning the development of the eyes. The eye development continues until after birth. It's not completed until after birth.
What is the black part of the brain?
Okay, I'll get there in a second. The ear development is also in this period of time. It extends for quite a period of time from about week 3 out through the first, essentially the first 16 weeks, so at least the first third of pregnancy. The palate and the teeth. And notice when external genitalia are developing, it's after you're into the 7th and 8th week before this is starting. The colored portion versus the black portion. The black portion indicates the period of time that is particularly sensitive to external influence for disruption of development. So recognize clinical recognition of pregnancy is here, and an awful lot of women would not know they were pregnant before about this range of time. So maybe they went out and had a party, okay, and they did some drinking, and maybe they smoked a joint or two, okay, or 3 or 4. And all of those chemicals, or they had a headache, and they took Tylenol or aspirin during this period of time. This, if they took enough of it, this can potentially affect development of the circulatory system and development of the central nervous system. I think you've all heard of alcohol-related deficits during development. Okay. The kids that have to go into tox programs after they're born. This is trying to indicate that there are sensitive periods for chemical exposures. Another common thing in the Western world, many young people are treated with retinoic acid to prevent acne, skin eruption. It turns out that retinoic acid is very, very toxic developmentally and can disrupt organ development in the embryo. Thalidomide. Anybody here heard of thalidomide before? What was it?
Didn't they, like, babies were born without, like, limbs and...
That was the end result. That's the end result.
Morning sickness or something like that?
Okay. What they were trying to do is they would give it to women who were complaining of morning sickness or who had had miscarriages, particularly repeated miscarriages. And they were attempting to support the pregnancy early on. Well, they may have supported mom, but what happened is it is a teratogen. It definitely affects developmental limbs. So although it may not have affected central nervous development, it did affect the arm and leg development very, very dramatically. But there's a lot of different chemicals that occur, not just in pharmaceuticals, but they may be nutrient supplements. They may be even things that you get normally in your daily diet. Caffeine at high levels is not good for pregnancy early on. All right. So the other thing I wanted to point to is the fact that sex determination occurs relatively late. It's out here at 7 and 8 weeks and beyond. Okay. So gonadal differentiation and development isn't really complete until somewhere about the 7th week of pregnancy, almost a third of the way through pregnancy. All right. Next. I'm not going to jump into sex determination quite yet. There's a couple of things I did want to bring up that were in other places. slide we were just on. This is showing you what happens in terms of the germ cell migration from, again, outside the embryo proper. It ends up migrating along the vessels leading into, and the structures leading into the hindgut. So these cells, these germ cell precursors, which are dividing actively by mitosis, migrate up the hindgut and locate themselves in what's called the genital ridge, which is the area on top or toward the head, and what will become the kidney. So the mesonephros is here, and there's the genital ridge. This is a cut. This would be this section showing you where those cells are located relative to one another. Got through that. All right. Into sex determination. Let me do something here first. Anybody here have a pet turtle? No pet, no turtle concerts? How about a fish? What kind of fish? Goldfish. What kind of fish? Betta fish. Koi. Fancy. Okay. What do you know about gender in fish? Nothing? There's males and females. You want to tell them about gender in fish?
Sure. I'm assuming you're going off what you say about turtles. So for turtles, it's temperature dependent.
Okay.
Fish depends on the species.
Okay. In fish, all right, in some species like turtles, and crocodilians, gender is determined by incubation temperature of the eggs. And you said in fish it depends on the species, and you're very right. Fish, you can get some fish that start as females and at some point become male due to developmental or social cues or environmental cues. You can also get some that start as males and become female. You know which ones you got? Yeah.
Why would temperature determine sex be advantageous? Because wouldn't temperature be the same throughout a large area? Would that mean everything in a large area would be the same sex?
You just put yourself into position to do a dissertation project. Nobody knows why it works that way. There's a suggestion that favoring one gender versus another is going to be protective of the species overall if the temperature is elevated. Okay. But yeah, nobody has fully figured that one out. Okay. Yeah.
Don't clownfish do one of those?
I believe so.
Sex, yeah.
I believe so. And there are some that actually will switch back and forth.
Yeah.
It's not just one direction.
Don't seahorses do that too?
I don't know about seahorses. Pardon?
It's the male.
But yes, Nemo does that.
Nemo.
Okay. So we don't know if he was finding dad or mom or. Okay. in mammals, how do we do it in mammals?
It's
How do
predetermined.
you get, how do you drive gender determination?
It's predetermined.
Pardon?
It's predetermined.
Predetermined by what?
Sperm cells.
Uh,
A higher power?
no, I mean,
Pardon?
Sperm.
Sperm?
Yes.
Y or X chromosome?
Well, whatever, either, Y or X carried by the sperm.
Okay, so you're saying it's chromosomally determined.
Yeah.
Partially.
You said partially.
Yeah.
Do you know something I don't?
Well, but you can have like SRY individuals.
Okay, okay. You're getting a little ahead of the game. But I asked the question, we'll actually touch on that. It's a very interesting story. Okay, it's not quite as simple as X and Y. Okay. But is this the whole story? So if this goes, this is always male.
No.
no.
Well,
No.
You can have X, Y females.
Pardon?
Have XY females.
You can have X, Y females.
Depends on how you're defining male and female.
Okay. That brings to another interesting point. It turns out that in mammals, sex determination
It's complicated.
is a process. It has multiple steps. And the end result can range from super male to super female. And anything in between. Okay?
That also depends on if you're looking at something genotypically versus phenotypically.
Yes, definitely. And we'll see why. Okay. It all comes down to this process business about determination. And that process is in large part determined not just by genetics, but by endocrinology. So, let's start talking about this interesting process. And I have a question to ask you at the end of it. We'll see if we get there today. Okay. So,
Get us excited.
I won't touch that today. Sex determination is a process. Okay. So, male XY chromosome with Y genes, XX chromosome with X genes. Okay. You've got genetic or gametic chromosomal sex determination. That's up here. And it turns out that a key feature of the XY system is the presence of a segment of the short arm of the Y chromosome that contains a small piece of DNA called SRY. It's also called the testis determining factor, or TDF. If this is present, it tends to drive the developmental program of the developing gonad toward the male tissue, that is, toward a testis. But not the full tissue. It's really only the central portion, the medullary portion of the what's called the indifferent gonad.
Professor?
Yeah.
Is that different from like a bipotential gonad? As I know zebrafish have like.
Well, it would be equivalent.
Okay.
Okay. Whoop, there we go. So the central portion particularly pushed toward the development of the cells that would normally occupy a testis. In the absence of this driver, this protein, this small protein that's expressed and drives the developmental program toward testicular development, you have the absence of SRY, and rather than getting the central portion of the indifferent gonad, you get the outer portion of the indifferent gonad to develop, and you move toward the formation of an ovary. This is not we used to think that this was a rather passive passage. It turns out that there are a number of proteins that are and factors that are triggered by the presence of SRY, and there are other factors that dominate in the presence or in the absence of SRY, which help drive this. So female differentiation is not absolutely passive, but it is largely passive. This is essentially the default pathway in the absence of SRY. Here's the trigger. Okay, yes?
So if in an XY chromosome individual, if they had no expression of the
SRY
protein, yes?
Well, it's actually, I believe it's a transcription factor.
Right. Would they develop female reproductive parts?
Yes, they would. They would develop, in fact, an ovary, which is part of the reason why we talk about this as a process, okay? And why you can get some very interesting steps in between the extremes. Yeah.
Regarding like someone who's a hermaphrodite, does that mean that the entire gonad is there?
Okay, in the purest form of hermaphrodism, you actually have testicular tissue and ovarian tissue in the same gonad, in the same individual. You may get it bilaterally or unilaterally. You can also see certain hermaphrodites that have an ovary on one side and a testis on the other, which is a hard one to figure out genetically. Yeah.
So looping what we learned before, if the transcription factor is not produced in XY individual, what about the other endocrine systems? LH production?
A lot of that is actually, will build on these stages. These are occurring beginning somewhere around 7 weeks and thereafter. At birth, you don't have a fully functional hypothalamic pituitary axis for a number of these systems.
So this comes first. And
This is one of the initial stages, yes.
Okay.
Okay, so we've got testicular development versus ovarian development. Now, one of the things that's going to happen in the testis is you're going to get development of early Sertoli cells, and those Sertoli cells are making a protein which is diffused into circulation, or at least into the area surrounding the organ, called Mullerian regression factor. It's also called anti-Mullerian hormone. It's got about 3 or 4 different names. So MRF equals anti-Mullerian hormone. As we go along, we may see several other descriptions of that. But this is a Mullerian regression factor. That's made by the Sertoli cells. It's not being pushed by any other hormone. It's being made by those cells early on. The Leydig cells, which are the other component of the testis, are being stimulated to begin production of testosterone, androgen. On the other side, you're not getting the ovary producing much in the way of steroids. Steroid levels remain pretty low. So you've got genetic or gametic sex we talked about from the chromosome. You have gonadal sex once you've started differentiating toward the ovary or the testis. And then you get into phenotypic sex. And the phenotypic sex has an internal component and an external component. So there are tissues interior that support the function of the ovary or the testis. And then externally is the external genitalia that goes along with one gender or the other. So testosterone will stimulate the persistence of an internal duct system called the Wolffian duct system. And the Wolffian duct system is going to develop into the vas deferens, the epididymis, the structures that support testicular function. The Mullerian regression factor, the Mullerian ductal system, is the tissue that would normally develop into the fallopian tubes in the uterus. In the presence of MRF, that regresses. Now, if things are going normally, there's also testosterone around at this time, so you've got the male duct system differentiating in response to testosterone, the female ductal system regressing in response to MRF. This acts locally. So the testis on one side causes regression, the Mullerian duct on that side, not on the other side. The testis on the other side causes ductal regression of the Mullerian ducts on the opposite side. Testosterone acts centrally, so it supports ductal development on both sides. Now, in the female, you don't have either of these things. And so, since you don't have testosterone to support Wolffian duct development, those ducts collapse, they involute, they undergo apoptosis. So no ducts for the support the male function. In addition, you don't have the regression factor. As a result, the Mullerian ducts develop. So you get internal, immature female genitalia. Notice I emphasize immature, because that's an important component of both of these. And the external sex characteristics in the female are going to be the vagina, the external labia, internal labia, the clitoris. Those are all going to be developing according to female phenotypic plan. But it's going to be immature at this stage. Likewise, the testis producing testosterone and MRF are going to stimulate male development toward an immature male internal phenotype with testosterone, with the testis and the supporting ductal structures and glands, accessory glands. And the male genitalia will form the immature penis, the scrotal compartment, which may or may not hold the testis at the time of birth. They sometimes descend after birth. And the testosterone is going to be converted by many of the external structures like the tissues of the penis, the prostate gland. Those tissues are going to convert this testosterone into an even more potent androgen called 5-alpha dihydrotestosterone. This is done by an enzyme called 5-alpha reductase, which forms this steroid. This is the real driver for male differentiation particularly in the external sex characteristics. It's not until puberty when the gonads actually stimulate production of androgens in the male, estradiol and progesterone in the female, that you get final differentiation of the external phenotype into an adult male, or an adult female. In addition to these goings on, you've also got these steroids driving, and even early on here in the male, these steroids driving the development of parts of the nervous system, parts of other organs like the liver and kidney, to follow a pattern that for their expressed behavior or metabolism that's gender specific. Because for instance, the enzymes in the liver of a male act differently from the enzymes in the liver of the female. There are often levels that have to be of expression that are involved. Yes?
If it seems like sex determination in a male at least is fairly linear, so one thing has to happen before another thing happens.
Right.
So why isn't sex determination for humans artificially affected? Like, if say a couple wanted a boy or a girl,
Right.
they just couldn't they either inhibit SRY production in order to have a girl, or to add SRY into fetal development to have a boy?
How do you propose to do that?
With a needle.
Placed where or when?
This is all fairly, we know what happens normally. Like you said, 7 weeks in,
Yeah.
SRY sex production. Yes.
Agreed.
So why don't you time it at 7 weeks yet? You know, jam the thing.
Jam the thing where?
In the you know, where the baby is born.
Oh my God.
No, does anybody have a problem with that?
Explain
Why?
why this doesn't work.
Well, first, I don't know how you would incorporate it into the genome of the embryo by just injecting.
I mean, the question I asked before that was, like, say an XY individual did not produce SRY, like would they develop female characteristics? And
You can
versus
do that in
yes?
rodents. You can introduce the opposite sex hormone and masculinize or demasculinize the brain.
But manipulating a human.
I'm not saying that's ethical.
I'm not saying I'm gonna go out with a needle with SRY in there. Correct, ladies. I'm just saying, I want to understand this, because this looks very linear and it looks very. It's probably not.
It
so,
is
but it
quite
looks very
linear.
simple.
Okay.
Yeah. So
Okay.
like, if I affect step 2,
Yeah.
it seems like it would affect step 3, 4, 5, and 6.
Right?
So why is
I'll
that?
give you even more in a minute. Okay. This is a small protein being expressed. Does anybody here know what antisense RNAs are? Never come across antisense RNAs. They are a commonly used tool in this day and age in biology. And I can make an RNA that's antisense to SRY. And I could potentially introduce it in an early stage embryo, but I'd have to have access to that embryo. Okay? Getting access to that embryo, once it's begun to implant into a uterine wall, is not easy. And it's not ethical. Okay? At this stage.
What do you mean at this stage?
So how do they do that?
Pardon?
Ethics are not defined by time.
Yes, they are. They certainly are.
Birth control would have been totally unethical 100 or 200 years ago.
True. How many test tube babies we got in the room?
Probably not yet.
I know one.
I know
Okay.
too, yeah.
I had a class 2 years ago, 15 students. There were 3 in vitro fertilization products in that room. Okay? It's not uncommon.
That's different than manipulating.
I don't know, is it though?
Is
Because
it?
some people feel like if you can't have kids, you can't have kids, but if you do it in vitro,
okay?
But you're shooting.
I mean, ultimately, what is that?
No, I'm not, because you ask about unethical issues, or issues being unethical. 50 years ago, it would have been questionable to do in vitro fertilization. Now,
It's totally normal.
one of the things that was almost blew me off my chair when I found out how many kids in the room were the products of IVF is that when we watched a movie about, it was actually a takeoff of a in vitro fertilization gone off the rails, essentially. It was called Delivery Man. Did anybody ever see that movie?
Yeah.
Okay. It's about this guy who was a sperm donor and ended up siring like 300 kids, okay? But he got connected to them, and it had a nice storyline to it anyway. And I'd shown it a couple of years before in a similar class, and it generated 2 days of discussion. In that class, it was kind of like, yeah, okay. Nothing. It was normal. So, yes, ethics change over time.
But I don't think that will change. Determining, I mean, it's like going to be a debate for a very long time if you can determine the sex of your offspring,
Like-
or if you can delete a gene for Huntington's disease or whatever.
We can do it with selective abortion right now,
Right.
okay?
Maybe there's a little country called China.
Our male-female population ain't very balanced right now.
Yeah. But yes, there technically, there is actually a way to interfere with it. Whether it's ethical to do that or how you would actually get in to do it would have to be worked out. And like I said,
But it's possible.
Technically, it's potentially possible. Whether or not it's going to be acceptable is a totally different thing. Okay.
You also, it may be early on, but if the SRY gene's already there, you would have to suppress it, right?
You would, absolutely.
Well, that's what you're doing with a inhibitory RNA.
Right, but you don't have that extra X chromosome that won't mess anything up.
Yeah,
No.
that's another one of my
Yeah,
questions.
this is what I'm getting to. This is the description of the research leading up to the identification of SRY as the sex-determining factor in mammals. And it wasn't really until 1959 that we recognized that the Y chromosome was in fact the determinant chromosome for gender determination in humans. And it wasn't until 1966, where they had identified the fact that the determinant factor was situated on this short arm of the Y chromosome. And as time went along, people were trying to identify where this region actually was, this subregion actually was on this short arm. In '86, somebody got very excited in New York because they thought they had the answer in the presence of this HY antigen. Okay. And there was a lot of excitement and research and talk about Nobel Prizes until they found out that there were individuals who existed that were HY positive but female. That kind of went out the window. So other investigators focus more on this region right here. Now it turns out that there's an interesting facet of the biochemistry, biology, physiology of this short arm of the Y chromosome. The distal end of the short arm of the Y chromosome turns out to be pseudoautosomal. It actually exchanges genetic information with the corresponding region of the X chromosome. There's actually a couple of places on the long arm of the Y chromosome that also do that. But this pseudoautosomal region meant that there was, Y chromosomes didn't just exist on their own. They actually could share genetic information with X chromosomes. And people got excited about this. HY was in that region. And, but they found out that there was this other region a little further out, a little closer to the pseudoautosomal region, that actually contained the sex determinant factor. And in '87, Dr. Page over at MIT, the Whitehead Institute, thought he had it. He found something called CFY, this particular region, which was just, was part of this segment, and they found it in all sorts of cases and gender crossovers that have been defined by physicians, and they found it in most, and all species they looked at until somebody said, well, have you tried it in marsupials? Marsupials are animals like kangaroos. They said, okay, well, we'll try it on kangaroos. Didn't work on kangaroos. Okay. So that ZFY was an interesting factor, but it turned out not to be the sex-determining region. And it wasn't until '89, McLaren and others identified this little segment, which was a high mobility group transcription factor that bound to the SRY element of Sertoli cells and Leydig cells. And that is the SRY gene. And when they isolated it and injected it into XX embryos early on, put those into foster mothers and allowed development, they got development of males that were XX, okay, but SRY positive. Yes?
This is a totally stupid question, but
No?
they say now that you can detect whether a woman is having a boy or a girl earlier.
Yeah.
Are they testing for SRY protein?
No, they're not testing for SRY. What they are doing is they're getting amniotic cells either out of her circulation or from the amniotic fluid. And they're doing XY determination, but also a suite of other genes.
Oh, okay, so they're just looking for genes.
Correct. They're looking for particular genes. So this was the critical set of experiments that demonstrated that SRY was crucial. And we'll finish this up next time, because I sense that we're moving out of the room.
You didn't ask the question.
Pardon?
You didn't ask the question.
Oh, that comes next time.
Oh, yeah.